Hi, my name is Varun Mangalick. Today we're going to talk about how DNA testing works and why it's so important in solving forensic cases. First, let's talk about a couple of the alternatives and why they're less than perfect. Let's start with eyewitness testimony. Studies show that people's memories can be influenced by their values, their expectations, and their cultural norms. Also, people are often just as confident in their inaccurate memories as they are in their accurate ones. What about fingerprint testing? That's supposed to be scientific, right? Well, most fingerprint analysis isn't done by some complex computer program. It's done by an expert staring at two fingerprints and seeing if they look similar enough. Also, studies show that not only are different experts often inconsistent with each other, a single expert can be inconsistent with themselves based on their mood or time of day. So what does work? DNA testing, and let's talk about how.
First, let's start with the basics. DNA is a type of molecule called a nucleic acid. It stores the cell's genetic information safe in a structure called the nucleus. DNA is made up of subunits called nucleotides. Nucleotides come in a few different varieties designated by A, T, C, and G. DNA structure is made up of two chains of nucleotides connected in such a way that A's and T's interact with each other and C's and G's interact with each other. This means when you're replicating DNA, you can just split the original two strands and build new strands following the pairing rules. Everyone has two sets of DNA that are slightly different in sequence, as one's from mom and the other from dad. This is cool and all, but what does DNA actually look like in real life without a microscope? Let's find out by extracting DNA from a strawberry. Let's check out all the materials we'll need. Water, salt, dish soap, isopropyl alcohol, cups, a couple of strawberries, plastic bags, a stirring stick or skewer, a plastic spoon, and a strainer. In laboratory DNA extractions, scientists use salts, detergents, and alcohols just like we're using here, but their chemicals work a bit better and are much harder to obtain. Strawberries are octoploid, meaning they have 8 instead of 2 sets of DNA, so we can be flexible with their materials and still extract enough DNA to see. First, put your isopropyl alcohol in the freezer. We'll get it later. Then, let's make our extraction solution. Add our 1/4-ish cup of water along with 1 tablespoon of dish soap and 1 teaspoon of salt to the extraction solution cup. Next, pour the solution along with the strawberries into a plastic bag. Try to get all the air out, then mash up that strawberry. Your strawberry demolition is helping your extraction solution get access to as many strawberry cells as possible in order to break down their membrane so the DNA comes out. Pour the solution through the sieve into the strawberry solution cup. Use your plastic spoon to really push as much liquid through as possible. Next, add about 1/4 cup of the isopropyl alcohol you had kept in the freezer. DNA floats in isopropyl alcohol unlike the rest of the strawberry gunk, so you can see at the top of your solution as wispy white strands. That's DNA. Now, let's move on to gel electrophoresis. Let's start out with our gel, a small slab of a squishy substance called agarose. If you looked at agarose under a microscope, it would look like a dense network of strands. Because of its molecular structure, DNA has a certain amount of negative charge per nucleotide. We can take advantage of this by putting the DNA at one end of the gel and putting a negative electrode on the DNA side and a positive electrode on the other. As DNA travels through the agarose, it gets caught in the matrix. Since bigger pieces get stuck more, they end up traveling more slowly down the gel. Now, let's say we started out with a bunch of DNA from a cell. Before running gel electrophoresis, we would probably want to cut up the DNA into smaller fragments since many of the chromosomes would be more than an inch long if uncoiled. The way this is done is with proteins called restriction enzymes. Each specific type of restriction enzyme has a certain 4 to 8 base pair long target sequence of DNA. If it sees that target sequence, it'll cut the DNA. A restriction digest is when you cut up a longer piece of DNA with one or more restriction enzymes. If you extract DNA from a cell, perform restriction digest on the entire extract, then run it with gel electrophoresis, you'd probably expect to get a gel with a few distinct bands where DNA traveled to. However, since cells have so much DNA and restriction enzymes cut reasonably frequently, you often just end up with so many different bands of different lengths that they all blend together. How do you get any information from this? What if we add a couple of steps to our procedure? Let's say you perform a restriction digest on 2 people's DNA using a restriction enzyme. Let's also say there's a certain gene on a DNA fragment in the restriction digest that you know you can learn valuable information from. If you know that gene contains a given target sequence, say ACTGAG, in all people, then you can find this band on the gel by using a nucleic acid probe, which is basically a short nucleic acid that can interact with your target sequence by base pairing rules and which glows with fluorescence, meaning we can shine invisible UV light on the probes and they'll emit visible light that we can see. You can bathe the gel from your gel electrophoresis procedure in a solution with your nucleic acid probe, then wash away the extra probe molecules. Then your target band will be glowing when you shine UV light on your gel.
So how can this help us solve forensic cases? Well, everyone has something in their genome called repetitive DNA. This is where a specific short sequence is repeated a certain number of times. While the specific short sequence is the same from one person to another, the number of times it's repeated is not. After you complete gel electrophoresis, you can use southern blotting with the probe targeting your short sequence to find the band with your repetitive DNA site on it. Then, based on how far the DNA band traveled on the gel, you can figure out the length of the fragment and accordingly the number of repeats in your repetitive DNA site. While two people might happen to have the same number of repeats at like site 3 and maybe site 6, they probably won't match at the multiple other sites for testing. So while some innocent bystander might happen to look superficially like the real criminal, the two individuals had different DNA, so that's something we can't mistake. This is just one simple example of the countless applications biotechnology has in fields ranging from forensics to medicine, agriculture, energy, and more. But that's all we have for today, so thank you for watching. Bye.